which will be from Gautier, Peyrat, and we'll talk about discovery of macrocyclic inhibitors of ALK using frags to drugs, a fragment-based drug design in silico tool. Gautier, it's up to you.
Thank you. So, hello everyone. First of all, thank you to JOBIM committee to give me the opportunity to present my work today. So I will present you the discovery of macrocyclic inhibitors of ALK using frags to drugs, a fragment-based drug design in silico tool. And I'm a PhD student of third year from the structural bioinformatics and cheminformatics team in Orléans. So what is fragment-based drug design? It is a method to discover inhibitors by using fragments, which are small molecules with molecular weights lower than 300 Daltons. Fragments have a low affinity but a high efficiency with their targets, and the three-dimensional information and structural biology are essential to perform a good fragment-based drug design. In the middle of the slide, I'll show you vemurafenib, which is the first inhibitor that was discovered from fragment-based drug design. In the study, the researcher went from a low activity of 200 micromolar for the first fragments to a better activity of lower than 20 nanomolar for the inhibitor vemurafenib. And the method that I'm using is fragment growing. It is how to extend one fragment to fit the target cavity by adding either several atoms or fragments. So in the method that I am using, I use frags to drugs, which is a fragment growing in silico tool. We developed and validated this tool on protein kinases, and its aim is to discover new protein kinase inhibitors from a 3D fragment network. It is available since few days at this URL, and you have also a QR code if you want to check the website that could where you can use it. This software is decomposed in three main parts. So the first is the graph database creation. Then we have the fragment growing, and then finally the selection of best molecules. So I said that it was developed on protein kinases. Protein kinases are proteins that phosphorylate other proteins. They are involved in various physiological mechanisms like cell survival, cellular transport, and cell signaling. And in human, there are 518 protein kinases. Any dysregulation or malfunction of those proteins can result in different pathologies, such as cancers, diabetes, or neurodegenerative diseases. Researchers have already found some inhibitors to inhibit protein kinases. And in our research team, we developed a database gathering all the protein kinase inhibitors that are currently in clinical trials or approved and on the market. So in the 1st of July, I counted 283 inhibitors that are in clinical trials. And among them, there are 69 inhibitors that were approved by the FDA. If we want to focus on the 3D structure of protein kinases, we can see that there are two main lobes, the N lobe and the C lobe. And those two lobes are linked by a hinge, which is also the place where the ATP, which is the natural ligand of protein kinases, is going to do its action. And in the, sorry, I've got my head on the right of the slide. In the catalytic site of protein kinases, there are some structural motifs and domains which are really conserved between all different protein kinases, such as the P loop, the DFG, or the activation loop. And those conserved motifs between all different protein kinases makes them hard to inhibit selectively, but also it gave us the entry point to create our database. So to create our database, we took all protein kinase ligands from the protein data bank. Then we superimposed all of them to have all the ligands in the same referential. Then we fragment all the ligands and we store them in a graph database where the nodes could be either a protein or fragment. And the edges are from three kinds. We can have compatibility relationship between a fragment and a protein, which will indicate that this fragment can go in the active site of this protein. And between several fragments, we can have inclusion or exclusion relationship, which will indicate if all the angles values and the distances between each fragment can allow to merge them in a molecule. So the fragment growing is performed by following all inclusion relationship in the graph. We start from one target and from one seed, which is the initial fragment. And by following the information of the graph database, we can grow to obtain several molecules until a certain molecular weight. And as you can see on the result that I show you in the right, all the molecules that are usually found by Frags2 drugs are molecules that are linear. And in this presentation, I want to improve this software to be able to discover macrocyclic inhibitors. So the selection of best molecule is the third part of the software. It is done in several steps. We apply some filters and some tags to be sure to find the best molecules. So we can calculate molecular descriptors to ensure that the molecule will correspond to protein kinase inhibitors. We can also calculate a synthetic accessibility score to be sure that our chemist colleagues will succeed to create the molecule, to synthesize the molecule. We can also perform some molecular docking to be sure that the molecule will form some good interaction with the target. And we can apply some tags like pan-assay interference patterns. This is some substructure that we can search in molecules to avoid some non-specific activity in biological assays. We can also search for similarity into known public database. This gives us two kinds of information. We can find some other molecules that are already active on other targets, or also we can check that the molecules that we have found are new because there are no similar molecules in public databases. And finally, we can order the molecule using a quantitative estimation of drug likeness, which is also a score that will indicate if the molecule is more or less drug-like. So the aim of this study is to discover macrocyclic inhibitors by using Frags2 drugs. We did a proof of concept on the protein anaplastic lymphoma kinase, so ALK. It is a tyrosine kinase involved in central nervous system. Any mutation of this protein can lead to several cancers, and there are already known inhibitors such as crizotinib, which is a linear inhibitor, and lorlatinib, which is a macrocyclic inhibitor, and the reason why we have chosen the protein ALK co-crystallides with the inhibitor lorlatinib. So macrocycles are molecules with a cycle of at least 12 atoms. They are able to inhibit challenging targets, and they can provide a better selectivity than other compounds on protein kinases. But they also have the drawback to be more difficult to synthesize than usual inhibitors. And software of fragment-based drug design are usually, it is hard to find macrocycles by using this kind of softwares. So I've chosen the PDB ID 4CLI, which contains the protein ALK complexed with lorlatinib. And in the inhibitor lorlatinib, I've selected one seed, which is the 2-aminopyridine. So it is squared in the top right corner. So from this target, this seed, and the graph database, I performed the growing, then I applied the filtering procedure to discover a new microcycle. And how could I find the microcycle? It is done just by following a path in the graph that will go from one node to other nodes and returning back to the first node. This will give us only molecule containing cycles. And we selected the macrocycles having at least 11 atoms and at most 15 atoms. So I applied this procedure and I discovered 592 macrocycles. Then I applied the filtering procedure and I went back to 153 macrocycles. Those 153 macrocycles were all new because they have no similar molecule in public database, which are Campbell, Ambinter, Zinc, and PKIDB. So you can see on the bottom of the slide examples of the macrocycles I found. And then I ordered the molecules according to their CAD score. So it is a score ranging from 0 to 1. The closer to 1 is the more drug-like molecule. So you can see here the 2D structure of the 9 best molecules. And on the right, you can see the 3D structures of the 153 inhibitors that are all superimposed. And among all the results that I got, there is one result which is really interesting because its 3D shape is really close to the inhibitor lorlatinib. So as you can see, the little cycles included in the macrocycle are in the same, almost the same place than lorlatinib. Even if the 2D similarity is only of 44% between the two 2D structures, we observe by just looking at the structures that the 3D shape is really similar. And also the CAD score of both molecules is also really close. So to conclude, I improved Frags2Drugs to be able to discover a new macrocycle. I did a proof of concept by discovering 153 macrocycles on ALK. And as a perspective, we want to synthesize the macrocycles that we have found and also to test them biologically. And we also want to continue this study by applying Frags2Drugs to discover macrocycles on new protein targets. So I would like to thank all the structural bioinformatics and cheminformatics team, and also especially all the researchers that contributed to this work, and you for your kind attention.
Thank you, Gautier, for your nice presentation. We have time for questions, so yes, you can ask them in the Q&A. So we have one first question from Sergei Godinin. How one can sample conformations of a microcycle?
I'm sorry, how?
How one can sample conformations of a microcycle?
What do you mean by sampling a conformation?
I guess because it's certainly a very constrained system. So how can you sample around the different conformations that this microcycle can have?
It is just by the fragments that we are using are just the fragments just collected from the protein data bank. So all the different fragments that we are adding in one microcycle are just coming from other ligands from protein data bank. So we don't search new samples. It is just one research from the information in the protein data bank.
Okay, but I guess the way you assemble the fragments, you can end up with different conformations, right?
Yes, only if the protein data bank had those conformations.
Okay. We have another question. So what software did you use for the docking step?
It is RDoc.
RDoc. Okay.
Yes.
Another question from Gwenaël André Leroux. What if you did not start from a 2-amino pyridine grain?
I guess the macrocycles would be different because the research in the graph would not end to the same 2-amino pyridine grain. So all the macrocycles would be different. I choose I will go back to the slide, but I choose this amino pyridine grain because it is the seed that is close to the hinge, which is the most important part in the interaction of inhibitors of protein kinases.
Okay. Another question from Stéphane Telechea. Can you tell us if the graph search can be constrained to another shape than a cavity, like for instance, the valley instead of a cavity inside the protein, so a pocket that is more open?
It could be if there are enough fragments from the protein data bank in this part of the protein. So we perform this in the cavity because there are a lot of fragments, so we can search some paths. We don't move the fragment at all. They stay in the position that they have in the protein data bank, and we just link them just by one bound, which can be simple, double, or triple. But we could do this in other parts of the protein only if there are enough information from the protein data bank.
Okay, but then in your docking stage, you might have it might end up in a different places, no?
Yes, but we do the docking only after the growing.
Ah.
So we grow, we obtain molecules, and then we just compare the structure that we got from the software Frag2Drugs, and we compare it to the docking of the same molecule, just to see if the molecule has not moved. And so we calculate the RMSD between both.
Okay. And I will take the last question. So you said that those macrocycles, they can be quite difficult to synthesize.
Yes.
Do you have a way to predict how difficult they would be to be synthesized? So it could be an additional filter.
Yes, actually we apply one score, which is the SA score. It is the synthetic accessibility score from RDKit. But since a few weeks, we have found a deep learning tool that is able to predict the routes of synthesis. So with macrocycle, this software has a lot of problems, but recently I sent to one chemist the routes from the deep learning software, and I hope that they will help us to synthesize the macrocycle.
Okay, great, thank you.
Thank you.
Thank you again for your presentation. We are on time for the next presentation from Sarah Nasseri, who I hope, yeah, she's here.